Hello, good afternoon, everybody. I hope everyone had a good break last week. Today, we will be going through lecture 5, which is about metabolism. We will go through the study guide. I'll do some summary of the lecture itself. And at the end, we'll have some time to go through the lab. So you probably know that the test is next Friday. I will go through some information about the tests when more people are here. Okay, so let's get started with the study guide. Okay, so this one you can get on eCentennial. Lecture 5, metabolism. First question, write the word equation and balance chemical equation for cellular respiration. Indicate which reactant is oxidized or reduced. So the word equation goes like this. It is glucose plus oxygen, and it's going to produce carbon dioxide and water. Now, in the process of breaking down glucose, we are going to release energy that can be captured by ATP. So ATP is adenosine triphosphate. The chemical equation is basically the chemical formulas for these molecules. So glucose is C6H12O6 plus oxygen, which is O2, carbon dioxide plus water, and up to 36 ATP molecules. So as you learn in chemistry class, you know you have to balance your equation. So we have 6 carbons here on the left, which means I need 6 carbon over here. And then there are 12 hydrogen in glucose. There are only 2 here, so I have to put in another 6. 6 times 2 gives me 12. And if you count all the oxygen, you will see that we are missing 12 on the left side. Okay, so that's a balanced chemical equation. Okay. So cellular respiration is actually a type of redox reaction. Redox reaction. So in redox reaction, it involves a transfer, of electrons. Okay, so some reactants are going to lose electron, other reactants are going to gain electrons. All right, now there are 2 sides to this then, right? There are oxidation and reduction. Okay, so oxidation means loss of electron, and then, or we would say losing electrons, and reduction means gaining electrons. Okay, so redox means like reduction and oxidation, right? Redox. You might have heard this before, right? Losing electron is oxidation. So people say LEO, losing electron, oxidation. And then gaining electron is reduction. So GER, LEO and GER. That's how you can remember which is which. Okay, so in our cellular respiration equation, the reactants, one of them is going to be oxidized, the other is going to be reduced. Can anybody tell me which one will be oxidized? Remember from the lecture recording, which one is oxidized? Paul said glucose, that would be correct. So glucose will be oxidized into carbon dioxide. Okay, so like this. A bunch of electrons will be removed from glucose. And at the end, you end up having carbon dioxide. So glucose is oxidized into carbon dioxide. And those electrons that you strip away from glucose will ultimately get to oxygen. So oxygen is going to be reduced into water, reduced. Okay, so that's basically what a redox reaction is. Any questions so far? Now, even though this is a single chemical reaction, right, glucose doesn't actually become carbon dioxide in one single step, right? There are actually many smaller steps in between, and we can break down the entire equation into 4 distinct phases. So I'm gonna review that with you. Okay, so here we have our cell, and then there is going to be the mitochondria here, right? With the cristae, and then this is the matrix, right? Matrix. This is cytoplasm. All right. So cellular respiration, the equation that we just looked at, that's the overall equation for cellular respiration. It is broken down into 4 phases, 4 stages. Okay? First stage is glycolysis. Glyco is sugar. Lysis is breaking. So you're breaking down the glucose. Okay? So that happens in the cytoplasm. Okay. Second step is the pyruvate oxidation, followed by Krebs cycle, also known as the citric acid cycle. Okay, so step 1, we're gonna take our glucose and we're gonna split it into 2 pyruvate. Okay. The pyruvate will then enter into the matrix where number 2 and 3 will take place. Sorry, it's really crowded here. Let me make it less crowded. Okay, so 2 and 3. And then finally, at the final stage, that's going to take place in the inner foldings. Oh boy, sorry. Let me try that one more time. There is the folding. Okay, so step number 4 is going to happen in the cristae, which is the foldings formed by the inner mitochondrial membrane. Electron transport chain. Okay. Now, during the first 3 steps, we are going to be stripping electrons from the glucose, okay? High energy electrons. So these are going to release high energy electron. Release high energy electrons. Okay, so something has to capture those electrons. What are the energy, what molecules are responsible for capturing the electrons? Can anybody tell me? My question is, as we remove electrons, these high energy electrons from the first 3 steps, something needs to capture those electrons. Otherwise, it's going to be wasted energy, right? So what's going to capture it? So Safi said FAD. That's one of them. Very good. And what's the other one? Anybody remember? Yeah. Jyoti said NAD. That's also correct. NAD plus. Very good. Okay. So these guys, these are energy carriers. Okay. Electron carriers. They capture the high energy electrons and then they will become, okay, so you add electrons to it, and hydrogen actually, but we focus on the electron. We'll capture the electron, and then the NAD plus is going to become NADH. And the FAD will become FADH2. Okay, so this is like empty with no electrons, and this is like fully charged with electrons. So how do I say this? How do I say this properly? Okay, listen to me. Well, I'll write down here, and then you tell me. NAD plus is blank into NADH. What's the word that I should put here? Is it oxidized or is it reduced? What is the correct word to put there? Please tell me. Is it oxidized into NADH or reduced into NADH? Anybody? Reduced is correct, right? Because it adds the electron to it, so it gains electron. That's why it's called reduced. Okay, same thing, right? For FAD, FAD is reduced into FADH2. Okay. Now, where are they taking the electrons? These things, the energy carrier, once they capture the electrons, they will take it to step number 4. Step number 4, like that. And then at step number 4, they will deposit the electrons. Okay, so the NADH will release the electron, so it will be oxidized back into NAD+. Same thing for the FADH2. It will release the electron and get oxidized back into FAD. And those electrons will be used to produce lots and lots of ATP in the last step, in the electron transport chain. Just to recap one more time very quickly. First 3 steps, we will be ripping electron from glucose. Okay, glucose is being oxidized. Okay, as we rip these electrons, high energy electrons from the first 3 steps, we have to capture the electrons. Otherwise, we will lose the energy. Okay, and that's no good. So to capture them, we need to have these energy carrier. Okay, so the energy carrier will capture the electrons, and then they become NADH and FADH2. They will transport the electrons to the final step, the ETC. They will unload the electron there, which will then be used to make ATP. Okay, so the NAD plus and FAD, they're like Uber. Okay, they're like taxis. They pick up passengers, drive them to the airport or something, and then they will go back, pick up more passengers, and drive them to the airport, back and forth all day long. Does that make sense? Any questions before we move on? Fantastic. Okay, now each of these steps have many, many mini steps, all right? And for the full glory detail, please watch the video. But I'm just gonna highlight some of the key things that happens for you, okay, as a review. So glycolysis. Okay, there are two major phases. Okay, we have the energy investing phase, and then we have the energy yielding phase. So in the energy investing phase, you actually have to use 2 ATPs, right, to energize the glucose. Okay, you give it a little bit of energy, you boost it, make it unstable so that it is going to react easier. Okay, so that's called the energy investing phase. It's like about 5 steps there. Okay, and then after you make the molecules unstable and energized, you are ready to harvest the energy. So in the energy yielding phase, step number 6 to 10, you will produce you will produce 4 ATPs. Okay. You will also be releasing electrons, right? And so those electrons will be captured by NAD, NAD plus. Okay. And so you will have 2 NAD plus coming, and they will become NADH. So you start with glucose, which is 6 carbon. At the end, you will split the glucose. You will split the glucose into 2 pyruvate. Pyruvate. Okay. Sometimes you hear people write pyruvic acid. It's the same thing. Okay. You guys learn from chemistry, right? Like acid has an H, right? So if you remove the H, then it becomes like ATE. Okay, like pyruvic acid, right? And then you remove the H, then we call it pyruvate. For us, it's the same meaning. It doesn't really matter. So help me out here. Glucose has 6 carbons. After you split them into pyruvate, how many carbons in each of the pyruvate? 3 is correct. Very good. So that is like in a nutshell, right? What glycolysis is. And this whole thing happens in the cytoplasm. So let's go back to our lovely chart here. We will fill it in. First step is glycolysis. And it is going to be taking place in the cytoplasm. Okay. So the key products will be 2 pyruvate, 2 ATP net. Okay, remember, you invested 2 and then you produce 4. So if you take away the 2 that you invested, you only make 2 net ATP. And then you're going to have 2 NADH. 2 NADH. Okay, those are the major products of glycolysis. Let's continue with our journey. So the second step is pyruvate oxidation. Right? As the name implies, you are going to oxidize pyruvate. What does that even mean? Well, to oxidize something means you take electrons from them, right? So you will remove more electrons from the pyruvate. Okay, so 3 things actually happens to our pyruvate here. Okay, number 1 is you will remove a carbon dioxide from it. It's called decarboxylation. It's in your PowerPoints. Okay, you remove a carbon dioxide. You will remove electrons from it, which then generates NADH. Okay, and then you're gonna add something to it. You're gonna add what's called a coenzyme. Coenzyme A. Okay, so coenzyme A, it's like a little booster molecule, right? It's gonna bind to our pyruvate and it's gonna charge it up so that it will be ready to react in the next step. Like the purpose of the coenzyme A is the exact same purpose as the 2 ATPs that we had in the energy investing phase. Okay, the 2 ATP is to energize the glucose, so it's going to react. Similarly, the coenzyme A is going to energize. It's going to create a special unstable bond with the pyruvate so that it is going to be ready to react. So then we're going to have something called acetyl-CoA. Help me out here. How many carbons do we have left from the original glucose? We started with 6 carbon in the glucose. At this point, how many of those original glucose will be left in 1 acetyl-CoA? These are important questions for the purpose of a test. Okay, so we have some people say 2. Anyone else? Do you agree it's 2? 1? Let's do a poll and see what everybody thinks. Go ahead. Okay, we'll say 1, 2, 3, 4. A, B, C, D. Okay, let's add in a 6 as well. Okay. The correct answer is 2. Okay, so you started with a glucose. Glucose has 6, right? 6 carbon. You split it into 2, so 3 and 3, right? That's the pyruvate. And then over here, you remove a carbon dioxide. One carbon dioxide has 1 carbon. So if pyruvate had 3 carbon, after removing 1 carbon from as in the form of carbon dioxide, you end up with 2, right? So that is the correct answer. Okay, so the exact same thing is going to happen with the other pyruvate. I'm going to copy and paste. So that is the pyruvate oxidation, right? What are the major products? We will have 2 carbon dioxide, 2 NADH, and 2 acetyl-CoA. Pyruvate oxidation. Where is it going to be happening? Matrix. Right? The matrix is the inside of the mitochondria, right? Matrix. And we will have 2 carbon dioxide, 2 NADH, and 2 acetyl-CoA. Those are the major products. Okay. Moving on. What's going to happen next? Right. So step 1, glycolysis. Step 2, pyruvate oxidation. Step 3, Krebs cycle. This is like a person's name, right? Name up to the person who discovered this, I suppose. Okay. So what's going to happen? Well, this is going to go and spin a cycle. This is also going to go and spin the cycle, right? So I'm just going to do one here. Now, where are we right now? Where is the acetyl-CoA? We are in the matrix, remember? So in the matrix, there is a molecule that's floating around. It is called oxaloacetate. Okay, big name, oxaloacetate. Now this oxaloacetate has 4 carbons in it. Okay. So the first thing that happens in the Krebs cycle is that this acetyl-CoA will combine with the oxaloacetate, and then it will create citric acid. It's the first product in the whole cycle. That's why it's also called the citric acid cycle. So again, help me out, folks. What is the number of carbon here? 1, 2, 3, 4, 5, 6. How many? Okay, Paul says 6. That would be correct because you have the 2. These are the original carbon, right, from the glucose, plus these random 4 carbon from oxaloacetate. 4 plus 2 gives you 6. Okay, and then what's going to happen is there are going to be like multiple steps, like a bunch of stuff is going to happen. We're not going to concern ourselves with what they are. If you took high school grade 12 biology, you would probably have to know what the steps are, but that's okay. We don't have to do that right now. And what you do have to know is you will be releasing 2 carbon dioxide, okay? And you will be releasing electrons, and then those will be captured by 3 NADH and 1 FADH2. Okay, and at the same time, you will also be producing 1 ATP. Okay, now stay with me here. We had the 2 original carbon, right, from the glucose. So I'm gonna put these as like red carbons. You can radioactively label them to track them. Okay, so 2 carbons. Those 2 carbons will now come out as carbon dioxide. Okay, so by the time the cycle is done, the original carbon from the glucose are all going to be released as carbon dioxide. Okay, so 2 plus 4 gives you the 6, and then 6 minus the 2 would bring you back to 4 carbons again. So those 4 carbons would be still in the oxaloacetate. So the cycle would regenerate oxaloacetate, and that's going to be ready to react again later on. Any questions so far? Okay, so this is going to happen. The exact same thing is going to happen for the other acetyl-CoA, right? For the other acetyl-CoA, like this. Right? So in total, for 1 glucose, the cycle will spin 2 times. Okay, so what are the major products? Let's write it down. We have the Krebs cycle. Krebs cycle, which is happening in the matrix. And we will get 4 carbon dioxide, 6 NADH, 2 FADH2, and then we will get 2 ATP as well. Okay, so by now we have generated the 6 carbon dioxide, right here, 6 carbon dioxide. And I know I said it already, but one more time. By the time we finish with the Krebs cycle, the entire glucose would have been fully oxidized into carbon dioxide. Okay, we are done ripping electrons from the glucose after Krebs cycle. Any question about anything at all? Okay, let me see if we can do these yet. Nope, not yet. Okay. No questions? Okay. Now let's take a small break here and talk about the test, and then we'll do more of this stuff. If you go on eCentennial, you will see that I have posted the test information. The test is going to be next Friday, October 29th. Is that 29th? Just double check. Yeah, October 29th. And I know some of you work, and you know, the previous time was not working out for everybody. So I extended the availability from 6 a.m. all the way to midnight, basically. All right, so you can do it before you go to work, or like when you end your late shift or something. The test will be 80 minutes, okay? And it will be lecture 2, 3, 4, 5, plus the lab. This is the link for the test. You can click on it on the day of the test. And here is a breakdown, right? 10 questions from lecture 2, 8 from 3, and 8 from 4. And then you have 20 from today's lecture. Okay, there are more for lecture 5 because you haven't been evaluated on it yet, right? Okay, 2, 3, 4, you did it on quiz 1 and 2 already. So there's going to be more questions for lecture 5. Beetroot lab, 4 multiple choice question. And then there are 10 application questions, mix and match from all these lectures. I decided to take away the multi-select questions for this test because those are the ones that people were struggling with in the previous quizzes. And the class average is not looking too well, right, so far with the with the quizzes. So I'm hoping by taking away the multi-select questions, it will boost the grades a little bit. Okay, they will come back later. Like for sure, I'll put them back in for later quizzes and tests. But for this test, I will take them away. It is going to be 20% of your final grade, 60 marks. Okay, so since the test is on Friday, we will have the test review on Tuesday. Okay, so next Tuesday is going to be our live session. Okay, and that is going to be like the regular time, like 12:30. Any question about the test, about the review, and you know, anything in general? I think you guys have like chemistry on Tuesday morning, right? Is that correct? So in the afternoon, you will have this you know, review. Please come for the review. Okay, review will be useful. After today's class, I will post the recording for this as usual, and then I will post a study package, review package for the test. You are encouraged to work on that before you come to the tutorial on Tuesday. I'll take up that review package on Tuesday. I will also post a practice quiz, like a multiple choice, like one of those self quizzes that some of you have been working on, right? As study tools. I'll post an additional one for the test. So you will have 3 attempts for that. It's not worth any marks, okay? But then you can do it as a review when you're ready, okay? So I'll provide the links to those after class. Okay, well, if there is no more question, let's go back to this thing here. Now, over here, I would like to talk a little bit more about how ATPs are being produced. Okay, so let me highlight the ATP. Right? In glycolysis, we are going to be producing 4 ATPs. Okay, no direct production of ATP from pyruvate oxidation, and then from Krebs cycle, we're gonna get, you know, another 2 ATP in total. So how do we produce those ATP? Okay, so there is a, oops, I don't know what I pressed. Hold on. There is sorry. There is a method, okay, a name for how it's being produced. So ATPs produce in glycolysis and Krebs cycle, or I should say, sorry, ATPs in glycolysis and Krebs cycles are produced by substrate level phosphorylation. Okay, so that's like a big word, but like what does that mean? Phosphorylation. To phosphorylate is to add a phosphate. Okay, so if you, e.g., phosphorylate ADP to become ATP. Okay. So ATP has 2 phosphate on it, diphosphate. And if you phosphorylate it, if you add a P to it, then it becomes ATP, triphosphate. Okay. So substrate level phosphorylation needs 3 things. And let me see if I have a picture for it in the PowerPoint. Okay. So we need 3 things. We need an enzyme, right? So an enzyme to catalyze a reaction. We need to have a fuel fuel or donor molecule. Okay, this is a molecule that has some phosphate on it, and we can remove that phosphate. And then we're going to need to have ADP. So what's going to happen is the enzyme will take the phosphate from the donor, okay? It will transfer it onto ADP, and afterwards you will generate ATP, and then the fuel molecule will have lost a phosphate because that phosphate went onto the ADP, right? So this is called substrate level phosphorylation, or SLP. You are phosphorylating on the substrate level, right? Like using an enzyme, basically, that's what they're saying. Okay. So not a lot of ATPs are produced this way, right? Only 4 out of the 36 ATPs that are produced is going to be using SLP. So that is approximately like 11.1% of all the ATPs. Everybody clear about what SLP means? Okay. Let's do some polls before we go too far. Okay, go ahead. That means the first 3 steps. You mean the SLP? Yeah. So SLP only happens in the first step and the 3rd step. Glycolysis and Krebs cycle. No ATP is produced in the pyruvate oxidation, right? So no SLP there. This is a tricky question, always on the test. A lot of people get it wrong usually, but hopefully not after I show you the answer. Okay. Let's share the results. The correct answer is pyruvate. Okay. Now, why is that? Remember, pyruvate is going to go on to pyruvate oxidation, step 2 and step 3. And in step 2, you will make 2 more NADH. In step 3, you will make, another 3 NADH, right? You will even make 1 ATP and then 1 FADH2, right? So all these things are going to have much more energy than any of these 3, right? So there's still a lot of energy locked in pyruvate, okay? At the end of glycolysis. Let's try the next one. This is a multi-select question. All right. Any last input? Okay. We just talked about this, right? From glycolysis and Krebs cycle. Right? 2 ATP are produced this way. Well, 4, okay, but 2 net from glycolysis, and then 2 ATPs per glucose from Krebs cycle. So A and C are the correct answers. Let's see this one. Okay, maybe we'll come back to this later. Okay, let's keep going with our chart here. The last part is going to be the electron transport chain, the ETC. Okay, so in ETC, it is going to happen in the cristae, which is the foldings created by the inner mitochondrial membrane. Okay, and the key products are going to be 32 ATP. Okay, this is where we cashed in on those high energy electrons. We're going to use those electrons to make lots and lots of ATP. So 32 in total. We will be regenerating the NAD plus, because after the NADH deposit the electrons, they're going to turn back to NAD plus, right? And then they can go back to the first 3 steps and get more electrons. Okay. And then we're going to have the FAD. We're not going to specify how many. We're just going to know that they will be regenerated. And we will also generate 6H2O. Okay. So that would be the 6H2O from here. Okay. So that is that. Let's go on to this chart here. I know I filled this, I think I filled this in the recordings, but I'm going to do it one more time just to help you get a grip on the math. So let's go through this again. Number of ATP produced through SLP, substrate level phosphorylation. So in glycolysis, we have 2 ATP produced this way, right? And again, I'm talking about the net. Okay, 2 net ATP produced this way. Energy carrier, okay, in glycolysis, and you can refer back to the first page, right? The chart that we just filled in the page above. The energy carrier produced would be 2 NADH. Okay. Now these 2 NADH is a little bit different than the rest of them. Okay. Here is the mitochondria again. Okay. This is the matrix. This is the cytoplasm. Okay. So the NADH that we produce in glycolysis, they actually cannot go into the matrix. Okay. And so what happens is they will just change back to NAD plus, and then they will pass the electron to FAD that is in the matrix, and then that's going to become FADH2. Okay, so the NADH does not go in, and then it gives the electron to the FAD. So the NADH here produced, it's actually going to become 2 FADH2. Okay, you lose a little bit of energy in this transfer. That's why it cannot be a NAD to an NAD, right? It's a NAD to a FAD. So for those of you who don't remember, each one NADH can produce up to 3 ATP, whereas one FADH2 can produce up to 2 ATP. So FADH2 has less energy than NADH. Okay, think of this as like 9-volt battery, and this is like a AAA. This one packs more energy. Okay, So let's take a look here. ATP produced at ETC, right? So these NADH will pass the electron to FADH2, and then we will have 2 FADH2 times 2 ATP, which makes 4 ATP in total. So from glycolysis, we directly produce 2, indirectly produce 4, so a total is 6 ATP. Okay, so ATP that is produced directly, we say substrate level phosphorylation. These ATP produced through the energy carrier, and we'll explain this more later, but it is called oxidative phosphorylation. Okay? Done. Pyruvate oxidation. Do we have any ATP from substrate level phosphorylation? If so, how many? That's right, there are none. Okay. Pyruvate oxidation only has 3 steps, right? You rip the carbon dioxide from it, you take some electrons from it, and then you add the coenzyme A, right? Okay. So because we're ripping electrons from it, we will produce 2 NADH. Now, these NADH will already be in the matrix, so no need to transfer them. Sorry, NADH. And so we have 2 NADH multiplied by 3. That creates 6 ATP. So in total, 6 ATP from pyruvate oxidation. Krebs cycle. Each glucose will spin the cycle twice. 1 spin, 1 ATP. 2 spins, 2 ATP. 6 NADH in total per glucose, 2 FADH2. And so please do the math here and tell me how many ATPs am I going to get? Anybody? Cynthia says 18. Anyone else? 14 from Michaela. Jyoti 22. Safi 12 and 4. Paul says 18. All right, let's find out. So 1 NADH can produce how many ATPs? Think about it in the mind. Is it 2 or is it 3? The answer is 3, right? So 6 NADH times 3 gives you 18. Each FADH2 will produce 2. So 2 times 2 is 4. So together, this is going to be 22 ATP, right? 22. Does that make sense? I hope it does. So all together, right, from Krebs cycle, 24. If you add them all up, 6 plus 6 is 12. 12 plus 24 is 36 ATP. Okay. Any question before we do polls? The number of ATP in glycolysis is 4, like in total, right? Or like in here, right? Total is 6 because we have to add the SLP as well, right? And then here is 4. Okay. All right. Poll time. Now let's do this one. All right, has been 1 minute. Just gonna give you a couple more seconds for the few of you who might still want to answer. Yeah, almost all of you got it right? The NADH has more chemical energy than FADH2 because each NADH can produce 3 ATP, but FADH2 can only produce 2, right? So B is the correct answer. This is not correct because NADH plus have to pick up the high energy electron to become NADH, right? So it's more energized afterwards. Okay, here is another one. Let's see if you guys can get it. Okay, a couple more seconds. So 4 NADH, so times 3, that's 12 ATP. FADH2 times 2, that's 6 ATP. If you add them together, 18 ATP. Okay, that's the correct answer. All right, next one. What is the maximum number of ATP that can be produced with 5 turns of the Krebs cycle? When you have the answer, please put it in the chat box. I should be clear, right? Like I'm asking for the number of ATP produced through like directly through SLP, as well as you have to think about the energy carrier going to the ETC as well, right? Okay, so not just the ones that you get directly. Does everybody understand what I mean by that? Like not just the ATP that is produced directly, but think of all the energy carrier that you will create, and then they go on to make ATP as well, right? Okay, so we have a lot of various numbers here, a lot of 10s, and 55 or 66. All right, let's take this up. So one cycle, one turn. What are the products of one turn? What's the product? What are the products from one spin? Okay. What's being pumped out? Okay. If you guys have been copying the notes in the tutorial, please look back to your notes and tell me, what do we get from one single turn? Anybody? Okay, let's go back to the notes. Where were we? You see, one turn will give you all these things, right? 1 ATP, 3 NADH, 1 FADH2. Okay. So one turn, 1 ATP by substrate level phosphorylation. And then we have 3 NADH and 1 FADH2. Is everybody clear on how I get these numbers? Because this is instrumental, right? To all the type of questions that are similar to this. Is that okay? Give me a thumbs up or like an okay, or ask a question. Okay. Okay, thank you, Paul. Thumbs up. Always appreciate the feedback. Okay, I assume the rest of you is okay then. All right. Now, what's gonna happen then? This stuff is gonna go on to the ETC, right? And then it's gonna create ATP. So I'm gonna multiply by 3 because each of the NADH can produce 3 ATP. So 3 times 3 is 9 ATP. This also will go on to the ETC, but it only has enough energy to produce 2 ATP. So that's going to be 2 ATP. So if you add all these up together, that is 12 ATP altogether for one turn. Okay. So if you turn this thing, if you spin this thing for 5 turns, 5 times, right? I'm going to multiply this by 5, which gives you 60 ATP. That's the answer. Does that make sense? And if not, please let me know which part is confusing. I'll be happy to explain it again. You will guarantee to see something like this on the test. Okay, which part, Christians, specifically you want me to explain? Like, do you understand these numbers? That's the first thing to understand. Does everybody understand where these things come from? Okay, so I mean, there isn't too much to explain here. It's just, it's based on what I've told you, right? The cycle, the Krebs cycle, maybe I'll show you the PowerPoint slide. Probably looks a little bit better. So like this is the actual Krebs cycle with many steps in it, right? But there is no way we're gonna memorize all that, right? So the simplified version is this. Okay, you're gonna have your acetyl CoA coming in, and it's going to combine with the molecules, and then it's going to go through a bunch of reactions. Okay, what's important is after all is said and done, the end result is you will get all these products. Okay, so you get this product, this product, this product, this product. So that's for one spin. So that's where I got all those numbers. 1 ATP, 3 NADH, 1 FADH2. Okay, where is my file? So that's how I got these numbers. It's basically like factual stuff. Okay, there is no, meaningful explanation behind it. It's the way it is. Okay, then these energy carrier will take the electrons to the ETC, and based on what I told you earlier, 1 NADH can produce up to 3 ATP. That's why if you have 3 NADH, it's going to be 3 times 3, which gives you the 9, right? And same thing for the FADH2, except we'll be multiplying by 2, right? So altogether, all these stuff is from one turn, right? So one turn alone will create 12 ATPs. And so if you do it 5 times, it's going to be 12 times 5. I hope that makes more sense. Any follow-up questions from anyone? Okay, I'm going to ask you a question. I hope all of you will get it right. Yes, one turn will always be the same. That is correct. Okay, this stuff is always the same for one turn. Okay, here is a question. Okay. Here is a question. Let's say I have 3 glucose. 3 glucose. How many ATP from Krebs cycle alone? Okay. Not all the steps, just Krebs cycles, right? Including ETC, of course. How many ATPs will be produced? Okay, from Krebs cycles. Okay, getting a lot of 72s. Anyone else? So Safi, Paul, and Michaela, you guys said 72. Any one of you want to explain? Want to unmute and explain, or you rather have me do the explaining? Anybody want to chime in? Okay, I will explain that. 1 glucose, how many spin? Yeah, so Safi said in the chat, 1 glucose has 2 Krebs cycles, right? Remember in the notes, right? 1 glucose split into 2 pyruvate, and then you will do the cycle 2 times, right? So if you have 3 glucose, sorry, I don't know where my stuffs are. If you have 3 glucose, how many turns is that? That's 6 turns, right? 6 turns. 1 turn will give you 12 ATP. So you would times it by 6 for this question. 6 times 12 is, of course, 72. Okay. I hope that makes sense. All right. Why don't we take a quick break, and then I will leave it on this page and let you, brew on it a little bit. We'll come back at 1:40, and then we'll do more. All right. See you soon. Let's continue. Okay. So, we are just gonna talk about like what exactly is happening in the ETC as a review, and then we'll move on to fermentation, and then we'll be done. So let's draw another picture. I encourage you to draw it with me. So this would be the outer membrane, outer mitochondrial membrane. And then we would have the inner membrane that folds into the cristae, right? Inner membrane, cristae. This space between the outer membrane and the inner membrane is the intermembrane space. And over here, that's the matrix. Right? Out here, cytoplasm. Right, so we're going to focus here. Now, embedded within the cristae is going to be a bunch of membrane proteins. So you can have a membrane protein here, a membrane protein here. You're going to have a membrane protein here. These membrane proteins, right? That's what forms the electron transport chain. So electron transport chain is nothing more than a bunch of membrane proteins that are like close together, form like a chain. Okay, so their job is to transport electron. That's why they're called electron transport chain. Right? So all those NADH that we got, all those FADH2 that we got, ultimately they will come to the ETC. So we have the NADH here. The NADH is going to release the electron, right? Once it releases the electron, it will turn back to NAD+. Okay, so the NADH will be oxidized into NAD+. And in the process, you are going to, I'm going to choose a color that you can actually see. In the process, you are going to release the electron. Okay. I don't know if you can see that. Oh boy. Sorry. It's beyond it's beyond fixation now. Here, maybe this will work. Electron. Okay, there we go. Electron. So it's going to release the electron, and then the electron will get passed down the ETC. Okay, like the hot potato game. You just pass the electron down the ETC. As you pass the high energy electron down the ETC, we will be harnessing the electron's energy. Okay, we'll be taking the energy from the electron and use it to pump H+ from the matrix into the intermembrane space. As this is happening, you will create high H+ concentration here. Okay, and then the inside the matrix will have low H+. And so as you learn in chemistry class, maybe high H+ means low pH. And this is going to be higher. Oh, sorry. High H+ means a low pH. Sorry, I was correct. Low pH means like acidic, right? And then if you have low H+, it means you're going to have higher pH. Okay. So all these H+, they really want to come back in. But they can't because they're a charged particle. And as we learned in lecture 4, charged particles cannot cross a membrane. So what do? Well, there is a special door for them to go through. Okay, it looks like a doorknob. It actually does. This is called the ATPase or ATP synthase. Both are acceptable terms depending where you read it. And so all these H+ that we pump over using the electron's energy will now go through the ATPase. Okay, so much like water going through a water wheel, it is going to turn the ATPase, which will help add a phosphate onto ADP, creating ATP. So that's how ATPs are generated from the electrons in the NAD+ and the FAD. Okay, so ATP produced this way is called oxidative phosphorylation, because the phosphorylation, the adding the P, is powered by oxidation reaction, right? Remember, the NADH is oxidized into NAD+, right? I keep on saying that. So that's why it's oxidative phosphorylation. Is that okay? Now, where does FADH fit into all this? Well, FADH2 does not have enough energy. And so it's going to release its electron at a lower point. Okay. So rather than pumping like 3H plus, it only has enough energy to pump like 2. So it does not have as much energy to produce ATP, right? So that's why NADH can produce 3 ATP, whereas FADH2 can only produce 2 ATPs. Okay, so that's essentially how the electron transport chain works. Now, there is also one final piece to this puzzle. Right? There is the involvement of oxygen. Oxygen is called the final electron acceptor. Okay, its job is to pull the electron from the last protein so that the next electron can slide down. Okay? Think of like what happens at the park. If you're a parent, you probably have seen this happen. Like my girls does this all the time. Okay, so like one of them slide down, but for some reason they don't want to leave at the bottom of the slide. So if they don't leave, the next person cannot slide down, right? So the parent has to come, okay, and then have to take the kid away from the slide so that the next one can come. Okay, so the parent is like the electron, right? It's like the oxygen, final electron acceptor. Okay, it's gonna pull that electron off the ETC so the next electron will slide down. When the oxygen pulls the electron out, it will form H2O. Remember in our overall cellular respiration equation, right? Oxygen is going to be reduced, right? Reduced means it's going to gain electron. So it gains the electron and then it's going to be reduced into water. And Christian said other kids would usually just crash into each other. Yeah, that's kind of true too. But that's a only analogy I can come up with. Okay, so that's what happens. Now, what happens if we don't have enough oxygen, if we don't breathe, okay? Then without the oxygen, the electron will not leave the protein. Then the electron cannot pass down. So the NADH has nowhere to deposit the electron. And then everything is clogged up. We cannot produce ATP. And you know, we're gonna die basically, okay, without oxygen. That's why we need it all the time. Does that make sense? Any question about what is going on here? Okay. Let's go back to the thing here. What is this process called? Oh, that's a tricky question. Yeah, the process of which the ATP is produced is called oxidative phosphorylation. Okay, the process of building up the H so that it will come back down. I don't know if I mentioned this word in the lecture. You guys can let me know if you have watched it. Chemia osmosis. Did I use this word or no? Did I? I did. Okay. Yeah. So that's basically what it is. Okay. The chemia osmosis is the fact that we build up the H plus, right, and then the H plus coming back in. Okay. So it's like osmosis, but it's with another chemical. In this case, H plus, and then how the ATP is produced is oxidative phosphorylation. The two are kind of interchangeable. Okay, if you would, for our purpose. Okay. Yeah. Let's go back to here. So, everything that I've talked about so far requires oxygen, right? So, you know, glucose becoming pyruvate. Okay, so we have two choices. Okay, O2 available, right? Then we're gonna go to like step 2, step 3, and step 4. Pyruvate oxidation, Krebs cycle, etc. And then if O2 not available, you're not gonna die just yet. We're gonna switch to fermentation. Okay. So all this stuff that we talked about up to this point requires the availability of oxygen. That's why we call it aerobic respiration. Okay. And fermentation, which I'll explain just right now, that occurs when you don't have enough oxygen, for humans anyways. So this is going to be anaerobic. Okay. So there are 2 types of fermentation. One of them is lactic acid fermentation. The other one is alcoholic fermentation. So let's go through them. Lactic acid fermentation is what happens in muscle cells, like our muscle. Right? If you are running at full speed, right, you cannot keep doing that forever. You're just going to be able to do it, I don't know, like 1 minute or 2 minutes, and then eventually going to have to slow down and stop. Right? So when you are running at full speed like that, eventually you're going to get to a point where oxygen demand from your muscle exceeds oxygen supply. Okay, then you're going to switch to lactic acid fermentation. Where is it going to happen in the cell? Cytoplasm. And then you're going to generate lactic acid. And you're going to regenerate NAD+. I'll explain in a second. Alcoholic fermentation happens for like things like yeast, bacteria. And we cannot do alcoholic fermentation because like we'd be drunk walking around, right? And alcohol is toxic to the cell. So when does it take place? Well, for these organisms, they rely on it. They do it all the time. All the time. Okay. Almost all the time. Cytoplasm. And they will produce carbon dioxide, which is why we can use yeast for baking, for making the dough rise. They also produce ethanol, which is drinking alcohol. The alcohol that we use to clean surfaces, right? That's not the same as drinking alcohol. Anybody know what those alcohol are called? The rubbing alcohols? What is that called? Anyone know? The one that you spray your grocery with. I don't know if anybody still do that. I don't. But what are those alcohol called? Anybody know? No, just trying to have a conversation here. Those are propanols. Okay, you cannot drink propanol. Don't drink it. You're gonna go blind and you're gonna die. Okay, so not a good thing. And then you're gonna generate NAD+ as well. Okay. So let's fill these in right here. Okay, so alcoholic fermentation. Now we started with glucose. We're gonna have glycolysis. Okay, you're gonna generate 2 ATP, and then you're gonna get 2 pyruvate. Okay, so that's not fermentation. That's just what happens normally. If you have enough oxygen, then the pyruvate will go on to do the next 3 steps: pyruvate oxidation, Krebs cycle, etc. Okay. Now, glycolysis also is going to release high energy electron. So the NAD+ will come, 2 of them will come, and you will produce NADH. Okay. Now, let me try to explain this a little bit more. Normally, when you have enough oxygen, the NADH will transfer the electron to the FADH in the matrix, right? Okay, but now remember, you don't have enough oxygen. The entire ETC is clogged up, so the NADH has nowhere to go. Okay, eventually you're going to use up all the NAD plus in the cytoplasm, and glycolysis will not be able to continue, and that's a big problem because remember, glycolysis has energy investing phase and energy yielding phase. So if you keep on investing and it does not yield, then we are going to be running on negative ATP, right? Okay, so that is very bad situation for the cell. What happens is then the pyruvate will, in the case of alcoholic fermentation, the pyruvate, it's going to release the carbon dioxide. That's what causes the bread to leaven. And then the product that you get is something called acid aldehyde. You will know what that means when you take organic chemistry next semester. And then the acid aldehyde is going to become the ethanol. Okay, so by doing this, you regenerate NAD plus so that glycolysis can keep on going and you will keep on creating 2 ATP. That's what happens in the yeast and the bacteria. In humans, it's quite similar. Okay, so glycolysis, okay, and then 2 NAD plus, 2 NADH. And then you will get the 2 pyruvate, right? Now, the pyruvate will become 2 lactic acid. Okay, so pyruvate is going to accept the electron from the NADH, and it's going to become lactic acid. So pyruvate is reduced into lactic acid. Okay, and the NADH is going to become NAD plus again. And the purpose of this is to allow you to keep glycolysis going. And so like you think about the full sprint, running super fast, okay, has been 2 minutes already, your legs are about to give up, you just almost can't do it anymore, right? Because you don't have enough oxygen. But this process, the lactic acid fermentation, will allow you to go an extra maybe 10 or 20 seconds, okay? But eventually you're gonna give up because these 2 ATPs that we're getting is just not enough to keep those muscles contracting, okay? And then how are you gonna feel the next day after like running full speed for as long as you can? Well, you're gonna have those burns in the muscle, right? So the lactic acid, people say it's what's causing the burn. Okay. Any question? No questions? All right. This part here, I am just gonna write the answers. I mean, I'll post this afterwards, and you can check it to see, and check for yourself. Okay, because we still have to take up the lab. So this is FIADEH. Okay, you don't have to copy it down. I'll post this. You can take a look after. Okay. The application questions. Okay, we can take a look here, and then we'll discuss what happens. Cyanide has a higher electronegativity than oxygen. Right, what does that mean? It means it attracts electrons better than oxygen. In cyanide poisoning, cyanide binds to one of the membrane proteins in the ETC. Explain how this could result in death very quickly. Okay, so let's go back to our picture of ETC. Okay, so oxygen is our final electron acceptor. Right, so I didn't really mention this explicitly in my explanation here. I might have in the recordings, I don't remember, but if you take a look at these electron transport proteins, right? Okay, we will call this protein A. I need a more standout color. We'll call this protein. Oh my goodness, so sorry. We will call this protein A, B, and C. Okay. Did you all learn about electronegativity in chemistry? I'm just wondering. Should I explain that, or did you learn that in chemistry already? Electronegativity. No? Okay. Electronegativity, EN. Okay, that basically means attraction for electrons. High electronegativity means high attraction. That's all it means. Okay. So if something has high electronegativity, it really wants to grab onto the electron. Okay. So let's take a look at our protein, our ABC protein, A, B, and C. Okay. We start with the electron here. Right? The electron is going to pass on to the next one and to the next one, and finally to oxygen. Final electron acceptor. Right? So in this order, which one has the highest electronegativity? Which one has the highest electronegativity? Well, actually, as you go down the chain, the next protein is the one that will have higher electronegativity, right? It has a higher attraction for electron. That's why it's able to rip the electron from it, right? B likes the electron more than A. That's why it will grab it from A. And C likes it more than B. That's why it will grab it from B. And oxygen will like it even more than C. And that's why it's going to grab it from them. Does that make sense? I think you can think of like, this is either going to help you or it's going to like even make it more confusing, but I'll try anyways. Think of like magnets, okay? The stronger the magnet, the easier it is to pick up a paper clip or something. Okay, so you have a bunch of magnets here. So you start with a paper clip here in the first magnet. How do you transfer the paper clip onto the next magnet? Is the next magnet going to be stronger or weaker? Stronger, right? Okay, so then this is a stronger magnet, so it sucks the paper clip from it, and then the next one is even stronger, it sucks it from it, and then so on and so forth. So as we go down the ETC, the electronegativity, right, is going to increase. Electronegativity is like the strength, the attraction for electron, kind of like the magnet, the attraction for the paper clip, except we're not trying to pull the paper clip, we're trying to pull the electron. Does that make sense? So back to our question. It says cyanide, right? CN, right? CN. Cyanide is a poison, and then it will get stuck. It will get stuck to one of the proteins. Okay? Never mind which one. It's just going to get stuck there. And the key thing here is that it says, where does it says? Cyanide has a higher electronegativity than oxygen. Okay? So what does that mean? It means when you introduce cyanide, that's the strongest magnet, right? Okay, so if you use that paperclip analogy, instead of passing a paperclip, normally oxygen will be able to pull it off from C, but in the presence of CN, because CN is now the strongest magnet, right? Oxygen no longer the strongest. The paperclip will be stuck to the CN, right? So in the same way, the electron will now be stuck to the cyanide. Oxygen will no longer be able to pull it down, right? So now you have all these electrons stuck to the ETC because the CN is stuck to the ETC. It's going to get clogged up, okay? It's like, you know, I don't know how to make this work for the analogy. It's like there's a sticky paper here and a bunch of kids is like stuck in the middle, okay? Now it's just all clogged up. Nobody can go, right? So we cannot deposit the electrons and we cannot make ATP. And yeah, we die. Okay, that's basically it. Any question? No questions. Okay. I will give you this question then. See if you can do it. Where is my poll button? Oh, there it is. These questions will tell me if you understood everything I said or not. Has been a minute. Only 2 answers. What about the rest of you? All right, I'm going to end the poll. And looks like we are split down the middle. Someone chose E, not sure why. So half of you say it's higher, half of you say it's lower. What's the correct answer? Does anybody want to justify their position? Anyone who chose A want to give me explanation? Or B, give me explanation. Okay. The ETC is clogged up, just like we explained, right? In the presence of cyanide, it's going to be clogged up. None of the electrons are going to go anywhere. They're just going to be stuck on the ETC. If the electrons are not moving, then can we still pump the H plus? No, right? If nothing's moving, we cannot pump the H plus. If we cannot pump the H plus into the intermembrane space, what's going to happen to the pH in the intermembrane space, right? We are going to have a lower pH, a lower concentration of H plus now, which means we're going to have a higher pH in the intermembrane space, right? What did the question ask? The pH to the intermembrane space. So it's going to be higher. The answer is A. Does that make sense? One more time. ETC clogged up because of the presence of CN. No ETC means you cannot pump the H plus. So the intermembrane space will have low H plus. Low H plus. What does low H plus mean? Higher pH, right? More basic as opposed to more acidic, right? So that's the answer. Okay, that's that. There are some fill in the blanks here. Maybe we'll take it up on Tuesday because now I need to take up the lab with you. Okay, here's the lab. Now, usually the lab is done pretty well. I mean, I have run this lab online since the beginning of the pandemic, right? So like I've done it how many times? One, two, like more than 6 times now, I think, and it always has been pretty good. But for some reason, this semester the average is not too good, and it's not just your class. It's across all these sessions. I'm not quite sure why. So if you think about some of the reasons why you found the lab to be challenging, okay, or if you have suggestions, please email me and I'll take that into consideration. Some of you already reached out and spoke to me, which is great. But I'm gonna go through the answers with you, and hopefully it will make sense. Okay, I'm just gonna verbally talk about it. Okay, so pay attention. What is the purpose of this experiment? I'm just looking for something very simple. Okay, the purpose of this experiment is to investigate the effects of temperature on the permeability of the membrane, right, of the beetroot membrane. That's all. Okay, no need to be too fancy. Next, write a hypothesis for this experiment. Some people wrote that you think temperature will affect the membrane permeability, but don't just tell me it will affect. You have to hypothesize how it is going to affect it, right? Okay, so an acceptable hypothesis would be the temperature will increase the permeability of the membrane, right? Or the temperature will decrease. As you increase temperature, you will decrease the permeability, right? Or you will say temperature has no effect, right? All those are acceptable hypothesis. It doesn't have to be correct. It just have to be like a prediction, right? Some of you wrote it as a question, right? Like some people put, does the temperature affects the permeability? Hypothesis should be a prediction, should be a statement, should not be a question. State the dependent and independent variable. So dependent is the one that you will be assessing, the one that you will be observing or measuring. In this case, we are observing the color intensity, right? The color intensity. Some of you put percent absorbance, which is fine. It's just another way to measure the intensity. But ultimately, right, whether you're doing it with eyes or you're doing it with an instrument, the dependent variable is the color intensity, right, of the solution. The independent variable is the one that you will be manipulating. So in this case, it is the temperature. Why is beetroot chosen for this experiment? So many people say because it has color, but it's 2 marks, right? So like you want to explain why not use carrot or tomatoes as well. So the beetroot, yes, the color is very important. The color is very easy to develop. It dissolves in the water, and you can tell the difference very easily. For carrot, why not use carrot? Well, carrot has the beta carotene, the orange pigment, but that doesn't really show up too well in the water. You might have 2 solutions. One have more carrot juice in it, so to speak. One doesn't, but then the comparison is not very obvious. Okay, so for visually obvious difference, that's why we choose the beetroot and not the carrot. The tomato is too watery. It's hard to cut them into precise pieces, right? And also the color does not develop too well either. So that's why we choose beetroot. Safety, I thought this was a pretty straightforward question, but I got a lot of like weird stuff. And many people just list a bunch of equipments here. I'm not sure why. Some people put like hot plates, sharp glasses. Like, what about them, right? So for hot plates, you don't want to be touching the surface. You want to always assume that it is hot, right? You want to tie your hair. If you have long hair, wear safety goggles, so on and so forth. Okay. So those are the safety precautions. This is another one that is surprising that not many people got. The control should be the one that is not manipulated, right? So 20 degrees, the room temperature one, that's your control, right? A lot of people put 0 as the control. Well, to get to 0, you kind of have to, chill the water, right? You have to put it in an ice bath, right? So that's manipulating it, right? So that's not the control. Why do we need a control in an experiment? Well, we need a control to compare the results with, right? If you see color in 40, 60, 80, and you don't have a control, how do you know that's not what normally would happen anyways? Okay, so by having a control, we know what the baseline is. We have something to compare our results with. Okay, so when we see a difference between the treated and the experimental group and the control group, right, we know is caused by the changes in the independent variable, in this case, the temperature. What needs to be taken into consideration? So the key thing I am looking for here is that we are trying to have a bunch of standardized variables, right? Remember in lecture 1, we talked about like experimenting with the cows, and I asked you, the group 1 cow and group 2 cows, what do you have to make sure that stays the same between the two groups? And people say like, you have to make sure that they're healthy, you have to make sure they're raised in the same environment, same age, that kind of stuff, right? So this is exactly the same question, but with regards to this experiment. So when you cut those core, you want them to be the same thickness, you want them to be the same size, you want them to be from the same plant, right? And why is this important, right? It's important because we want to make sure that any difference we observe later on is not due to inconsistency of the beetroot pieces themselves, but caused by the change in the temperature. Okay. Why is it necessary to rinse the cut beetroots? Almost everybody got this because when you cut them, you damage the cells. You want to rinse away the excess pigment that has been leaked out. This is another tough one that another one that a lot of people got wrong, I should say, but it's not that tough. Why is it important to leave the beetroot in the test tube for 20 minutes, not longer, not shorter? Okay. The reason is this, very simple. If you leave them in for too short, let's say you do it for 5 minutes, you might not have sufficient time to wait for the diffusion to take place, right? So you might end up having a bunch of tubes that are all pale colors. Okay, 40, 60, 80, they all look pale. If you leave them in too long, then you leave too much time for the diffusion to take place, then they might all look very dark. Okay, so 20 is kind of like the time where you leave just enough time for the diffusion to occur, and you can still observe a significant difference. Okay, so some people explain that quite well. Others kind of miss the point, but that's the answer. Okay, correlation. Right? It's only 1 mark. Always look at how many marks it's worth, and then you kind of think about how much you have to say. Some people roll a lot of stuff to describe this situation, but all I'm looking for is that the color intensity is higher at higher temperature. That's what you see, right? 80 is really dark, and then it's lighter as you go to the cooler temperature. That's all I'm looking for. Right? More intense color at higher temperature. The answer you gave in part A is an example of qualitative analysis. Explain what this means. Many people copy a definition from somewhere in the internet about what qualitative analysis is, but it's basically what we talked about in lecture 1, right? Qualitative analysis is an observation that we make using our senses. And in this case, we are using sight, right? To see the color intensity. Okay, so darker color, lighter color, right? It's a subjective thing, right? Because we're not using actual numbers. So quantitatively, right? The person use a colorimeter, right? A device to measure color intensity. I call it a spectrophotometer. You can say either, you will get the full mark. Relationship between absorbance measured by the spectrophotometer and the color intensity. Well, the higher the intensity, the higher the absorbance. Okay, so if you have a tube of red solution, it will absorb all the color except for red. That's why it appears to be red, right? So when the light strikes the tube, and then everything is absorbed, but the red is reflected and it goes into your eyes. That's why you see it as red, right? So when you shine green light at it, how much of the green light is absorbed, right? That's going to be determined by how much red pigment you have. So higher color intensity means higher absorbance. This one is, I was also really surprised. Usually This one, I expect people to get full marks, but many of you did not label the axes, the x-axis and the y-axis. It says your graph should be properly labeled, right? With the axes and everything. So x-axis is temperature, y-axis is percent absorbance. And if you watch the video, you would notice the person used a line graph. So I got all sorts of real graph. Some people use a pie chart for this, which is not an appropriate way of representing the data, right? Okay, so line graph. Bar graph is okay, not the best, but I still accepted the bar graph. But most people lose mark because they did not label the axes. Okay, and also some people instead of pasting the graph here, they put a link to some other location. They put the file on like a Dropbox or something, and then I wasn't able to open it in some cases. So you wouldn't get a mark if you do that. So for the next lab, the microscopy lab, you will have to copy and paste a lot of image onto the lab. So make sure you just paste the image. Okay, don't give me a link that I have to open because God knows why it wouldn't open. Okay, so that's that. And I'm not necessarily talking about anybody who's here, but people who are watching this tutorial recording. If that's the case, make sure next time you learn how to do copy and paste. Okay, last part. At what temperature does the absorbance peak? 60 degree. Right? After that, it kind of plateaus a little bit. And do you expect this to be the same for other types of membrane? Many people say yes, but the answer is actually no, because the permeability is going to be dependent on the composition of the cell membrane. No two cell membrane from different organism is going to be identical, right? The cabbage might have more proteins in it, and therefore it's more resilient to temperature compared to the beetroot, okay, or vice versa. So if the composition is different, you would expect the peak temperature to be different, okay? Provide two reasons explaining how temperature affects membrane permeability. This was described in the video. Between the 40 and the 60 zone, the temperature causes the phospholipids to move a little bit faster, okay? And then when they move faster, that creates more fluidity, and then things are able to leak past them easier. And then after 60, that's where the protein starts to denature. Okay, once the proteins denature, then the entire membrane integrity is going to be compromised, and then that's where you have a huge leak in pigments. So those are two reasons: increase movement from the phospholipids at a relatively low temperature, and then at high temperature, denaturation of the protein. Conclusion, and whether you accept or not, all you have to say is higher temperature increases the permeability of the membrane, right? Because you observe a higher color intensity, and you have to tell me if that agrees with your hypothesis or not. So you either accept it or you reject it, depending on what you predicted initially. Number 4 was a disaster. I actually went through this in tutorial number 4. Okay, for those of you who attend, you would probably remember I talked about this, and you probably got it right on the lab. Okay, in the tutorial, I asked you what happens if a piece of frozen cell is placed in regular water, right? Okay, and I told you when you freeze the cell, the ice crystal is going to damage the cell membrane. Okay, so if you freeze the beetroot, the whole cell membrane is like full of holes. So when you put it back in room temperature, it doesn't even matter. It doesn't even matter what temperature you put it in, because the entire membrane is full of holes. So it's just going to leak all over the place, regardless of what temperature you put it in, right? That's why, even though you put it at room temperature, you would have an absorbance that is 84, really, really high, right? Similar to what you would observe at 60 or 80. So that's the reason. Okay, so that's the lab, and there's only 4 questions related to the lab on the test. So if you understood what I just said, you should be okay. And that's it for today. Okay, I will post all the information that you need, and I'll see you all on Tuesday. Okay, have a good weekend. Bye for now.